Potato blight, also known as late blight, is caused by the microorganism Phytophthora infestans. It was first discovered in the United States in 1843. The disease is most famously associated with the Irish Potato Famine of the mid-19th century, which began in 1845 when the blight ravaged potato crops across Ireland, leading to widespread famine and a significant loss of life. This period marks the first widespread and devastating outbreak of potato blight in history. The root of potato blight infection, caused by the pathogen Phytophthora infestans, involves a complex interaction between the microorganism, the potato plant, and environmental conditions. The infection process begins with the dispersal of fungal spores, which can be carried by the wind, rain, or cling to soil and farming equipment. These spores are highly adaptable and capable of traveling long distances under suitable conditions. Upon landing on a potato plant, these spores require moisture to germinate, a process facilitated by high humidity or wet conditions persisting for several hours. This leads to the production of zoospores, motile spores that swim in water films to find an entry point into the plant. Once an entry point is found, the zoospores insist and germinate, producing structures that penetrate the plant's tissues, marking the beginning of the infection. The visible symptoms of the infection manifest as water-soaked lesions on the leaves, which quickly expand into larger, brownish-black areas, indicating extensive tissue damage. The disease also affects the stems and significantly the tubers, which develop dark, rotten areas, compromising their quality and edibility. As the disease progresses, the fungus thrives under cool, moist conditions, producing more spores on the infected plant parts. These spores serve as a means to infect neighboring plants, propagating the disease throughout the potato field. Moreover, the pathogen can spread to the tubers, either directly from the soil where spores infect the tuber surface or through the plant's stem, leading to the tuber's rot either in the ground or during storage. A critical aspect of the potato blight's life cycle is its ability to overwinter on infected plant debris or within infected tubers left in the field or stored improperly. These infected tubers then become the initial source of infection for the next planting season, perpetuating the cycle of the disease. Environmental conditions, particularly wet and cool weather, play a significant role in facilitating the spread and severity of potato blight. Effective management strategies, including crop rotation, the use of resistant potato varieties, and the timely application of fungicides, are crucial in controlling the outbreak and spread of this devastating plant disease. Potato blight, caused by the pathogen Phytophthora infestans, presents a series of distinctive symptoms on potato plants, affecting both the foliage and the tubers, which are crucial for early detection and management of the disease. The symptoms manifest initially on the foliage as small, dark, water-soaked spots. These spots can rapidly enlarge under favorable conditions, such as cool and moist environments, turning into larger, brownish-black lesions. Around these lesions, the leaf may show signs of yellowing, indicating the plant's stress and damage. A characteristic feature of these lesions, especially under wet conditions, is the presence of a fuzzy white growth on the edges, which is the spore-producing structure of the pathogen. As the disease progresses, the infected leaves may wilt, die, and the plant can lose leaves prematurely. This reduction in foliage not only affects the plant's ability to photosynthesize but also its overall vitality. The stems of the potato plants can also be affected, developing dark, elongated lesions that weaken the stem structure and can lead to the plant collapsing. When it comes to the tubers, which are of significant agricultural and economic importance, the symptoms include dark, sunken spots on their surface. Cutting into these tubers reveals a reddish-brown discoloration extending into the flesh, a sign of the pathogen's presence. If secondary bacteria invade these tissues, the infected tuber's flesh can become soft and rotten, often accompanied by a foul odor. These symptoms on the tubers can arise from direct contact with spores in the soil or from the pathogen moving down the stem into the tuber itself. The overall impact of the disease on infected plants includes stunted growth and a reduction in yield. In severe cases, entire fields can be devastated, significantly affecting the quality and quantity of the harvested tubers. Recognizing and managing the symptoms of potato blight involves a comprehensive approach, including the use of resistant varieties, implementing crop rotation, monitoring environmental conditions conducive to the disease's spread, and applying fungicides when necessary. Early detection and prompt action are essential to mitigate the impact of this devastating plant disease. Treating potato blight, a disease caused by Phytophthora infestans, requires a multifaceted approach that combines cultural practices, chemical interventions, and the cultivation of resistant varieties to manage and mitigate its impact effectively. An integrated management strategy is crucial for controlling this disease, which can devastate potato crops. Cultural practices play a foundational role in blight management. Implementing crop rotation helps prevent the accumulation of pathogens in the soil, as continuously planting potatoes or other susceptible crops in the same area can increase disease pressure. Proper plant spacing is essential to enhance air circulation among the plants, reducing the humidity that facilitates the disease's spread. Effective water management practices, such as watering plants in the morning and favoring drip irrigation over overhead watering, are critical in keeping foliage dry and less susceptible to infection. Regular monitoring of the crop to identify and remove infected plants promptly can prevent the spread of blight to healthy plants. Additionally, thorough sanitation measures, including the removal and disposal of plant debris at the season's end, are vital to eliminating potential sources of infection for the following year. Chemical treatments, particularly the use of fungicides, are often necessary to protect crops from potato blight. The strategic application of fungicides, based on weather conditions and the crop's growth stage, can significantly reduce disease incidence. It is generally advised to begin fungicide applications before the disease appears, especially under conditions favorable to blight development. The selection and rotation of fungicides with different modes of action are critical to prevent the pathogen from developing resistance. Protective fungicides are used to prevent the disease, while curative fungicides are applied when the disease is detected, though they are less effective in reversing damage. The cultivation of potato varieties resistant to blight is another cornerstone of an effective management strategy. While no variety is completely immune to the disease, resistant cultivars can significantly reduce the severity of outbreaks. Using certified seed potatoes that are free from disease ensures a healthy start to the crop. Environmental and monitoring strategies, including the use of blight forecasting systems, can inform the timing of fungicide applications, enhancing their effectiveness. Maintaining field hygiene by keeping field edges mowed and removing volunteer potato plants also helps in reducing the risk of disease spread. Delaying the harvest until the foliage has completely died back can further minimize the risk of tuber infection. An integrated pest management (IPM) approach, incorporating these diverse strategies, is the most effective way to manage potato blight. By staying informed about the latest research and advances in disease management, farmers can adapt their practices to protect their crops from this destructive disease, ensuring healthy growth and minimizing yield losses.